Teshthile, welcome to class 12 biology. In this video we are going to discuss about linkage and recombination. Linkage and recombination were observed by Thomas Hunt Morgan and his colleagues while they were carrying out experiments with Drosophila melanogaster. If you recall, in my last video I told you that Thomas Hunt Morgan was the person who confirmed the chromosomal theory of inheritance, which was proposed by Sutton and Boveri. So how did he confirm the chromosomal theory of inheritance? I didn't explain you properly. I thought I will explain that before we move forward with linkage and recombination. So Thomas Hunt Morgan, he was able to confirm the chromosomal theory of inheritance by carrying out a monohybrid cross between two Drosophila which differed in their eye color. Important thing to keep in mind over here is that normal Drosophilas, the wild type Drosophilas, they have got red eye color. The mutant types, they will have white eye color. So in his monohybrid cross, what he did was he took a red-eyed female. So this symbol remains denotes female. A red-eyed female was crossed with a white-eyed male. So this symbol is for male. So a red-eyed female was crossed with a white-eyed male, and he obtained the F1 individuals. And all the F1 individuals, let it be male or female, they were with red eyes. So he was able to tell that the red eye trait was dominant over the white eye trait. And when he carried his experiment forward, he crossed two F1 individuals or two F1 hybrids. He obtained F2 generation. So in the F2 generation, he obtained a typical Mendelian monohybrid phenotypic ratio, that is 3:1, in which the 3/4 of the F2 individuals were with red eyes, and 1/4 of the F2 individuals were with white eyes. But a little bit difference were there. In Morgan's cross, the F2 individuals, the ones which were having white eyes, they were all males. There were no females which were having white eyes. So how did he explain that? So he assumed that the genes for the eye color, were located on the X chromosomes. So when so he also knew that the females, they have got two X chromosomes, and the males, they have got one X chromosome and one Y chromosome. So he wrote down the genotypes just like this. So when he wrote down the genotypes, he was able to obtain the exact result that he obtained through his experiment with Drosophila melanogaster, and through that way he was able to explain that the genes were located on chromosomes, and therefore the chromosomal theory of inheritance was correct. The important thing over here is the representation of the genotypes. I just represented the genotypes like this, but in most of the textbooks and reference books you will find the genotypic representation in this particular format, whereas the W plus X, W plus denotes the wild type, which is the red eye color, whereas XW, which denotes white eye color. So this is the mutant type. So this is just for your awareness, that you have to remember, if you are given this particular genotypic representation, you have to know that this particular individual is female because it has got two X chromosomes, and since there are W plus written over here, it is homozygous for red eye color. So this is the wild type. And if you are given a genotypic representation like this, you have to know that this is for male because it has got one X chromosome and one Y chromosome, and the W represents the X is carrying white eye color. So over here he said that the chromosomes, the X chromosomes, were the ones which were carrying the gene for the eye color. With this monohybrid cross, Morgan was able to confirm the chromosomal theory of inheritance. I hope you understood how he was able to do that. So now let's move on to the main topic of this video, that is linkage and recombination. Like I said before, linkage and recombination were observed by Morgan. How did he observe that? He observed that when he was carrying out his dihybrid crosses with Drosophilas. In the dihybrid crosses, we know that the parental individuals, they differ in two characters. Over here, we have taken two parents which differs in two characters, that is body color and eye color. So when Morgan carried out cross between two such individuals, he got F2 ratio which deviated significantly from the Mendel's 9:3:3:1. What does that mean? That means that the genes responsible for the body color and the eye color, they did not segregate independently during gamete formation. So during gamete formation, the gene responsible for the body color and eye color, they did not segregate independently, and therefore the Morgan's F2 ratio was not in 9:3:3:1. What he saw was the parental combinations were much more in number compared to the recombinant ones, the new character combinations. So how did he explain this particular phenomenon? He said that the genes responsible for the body color and the eye color must be located on the same chromosome. This physical association between the two genes interfered with the independent segregation of the alleles. So let's look at this one over here. When there are two genes and these two genes are located on different chromosomes, when the two genes are located on different chromosomes, they are said to be unlinked. Over here, the gene A and gene B, they are not linked, they are unlinked genes. And when they are not linked, what we get is such kind of dihybrid crosses. What we will get is, we will get the Mendelian cross, that is Mendelian ratio, that is 9:3:3:1 dihybrid ratio. How do we get that? Let's take a look over here. Let's take a look at this particular picture. When gene A and gene B are located on different chromosomes, when gene A and gene B are located on different chromosomes, during gamete formation, during meiosis, what happens is they assort themselves, or they segregate in such a way that the segregation of A is independent of the segregation of gene B, and therefore we get four different types of gametes, each in equal proportions, that is 25%, 25%, 25%, and 25%. So we get four different gametes in equal proportions, and because of that we get the combination, or the F2 ratio of 9:3:3:1. Now let's see another case in which the gene A and gene B are located on the same chromosome. When the gene A and gene B are located on the same chromosome, they are said to be linked genes. So when the genes are linked, and if we carry out a dihybrid cross, what we observe is we do not get the 9:3:3:1 F2 ratio. Why don't we observe that? We don't observe that because when the genes are located close to each other, when the two genes are located close to each other, the chances of A and B getting segregated, this capital A and this capital B getting segregated or separated, is very less. This small a and small b getting separated, the chances of getting them getting separated is very less, and therefore we observe more of them in number. Over here in the gametes, we find that the capital A and capital B, they are more in number. The small a and the small b, they are more in number, 48%, 48%. Whereas the recombinant ones, right, capital A segregating along with a small b is very less, only 2%. Small a segregating with capital B, that is also 2%. So these two are called as recombinant. So the recombinant numbers are very less compared to the parental gene combination. So this happens due to these two genes being closely linked on the same chromosome, being in close position with each other. So this is called as linkage, right? And among the linked genes, there are two different types. Let's look at this particular picture. When gene A and gene B, let's look over here. The gene A and gene B, they are very close to each other as compared to gene A and gene C, right? So gene A and gene B, they are located very close to each other, so they are said to be tightly linked. Whereas gene A and gene C, since they are far away from each other, they are said to be loosely linked. The closer the genes, there is less chance of them getting separated in the gamete formation, during gamete formation. Further, the genes, when the genes are loosely linked and when they are far away from each other, there is a higher chance of them getting segregated, and they get segregated because of this particular phenomenon, that is recombination. You must have studied about recombination in your class 11. How does recombination happen? When the homologous pair of chromosomes, when they align themselves at the metaphasic plate, what happens is they exchange material with each other. The non-sister chromatids, they exchange chromosomal material with each other. So look over here, the blue chromosome, it is giving its part, right, one part of its arm to the red chromosome, and the red chromosome is giving one part of its arm, chromosomal arm, to the blue one. So this is called as recombination, right? So this is the recombination process happening over here. Now let's see, if on the chromosomes we have got these genes, right? The blue one is carrying all the dominant genes and the red one is carrying all the recessive genes. So how does recombination happen? When the recombination happens, when the crossing over happens, when the chromosomal arm overlaps over each other and then there is an exchange of materials, what happens is the recombination happens. Remember, earlier all the dominant alleles were present on the blue one, but now let's look over here. The recessive C, right? The recessive C allele is now on the blue one, along with the blue one, right? So this shows recombination. So this is called as recombination, and recombination is the main phenomenon which leads to the variation among sexually reproducing organisms. So if you have understood these things, then I think this will make sense to you now. This particular picture you can find in your textbook, and this picture is really important. It may come in your examination as well. I have seen board exam questions which has asked you to explain this particular picture, right? What does this particular picture mean? This particular picture simply means that when the two genes are located close to each other, right, the recombinant types in the F2 generation will be very less. See, when the Y and W, which means the yellow body and white eye color genes, right, they are located very close to each other. The body color and the eye color, they are located very close to each other, and because they are tightly linked, the recombinant types are very less in number. That is only 1.3% recombinant types are there. The recombination frequency is just 1.3%, whereas the parental types, right, the yellow, white and wild types are much more in number in F2 generation. So this is tightly linked. But when the genes are loosely linked, when they are far away, even though they are on the same chromosome, when they are far away, what happens is the chances of recombinant types, the chances of the formation of recombinant types is much higher. It is 37.2%. Right? When the genes are located further away on the same chromosome, the chances of recombination, right, the overlapping of the chromosomes, the crossing over, right, there's more chances that W and M, right, white and miniature genes, they get segregated independently, right, and therefore more recombinants are formed. The recombination frequency is 37.2%. It shows that the W and M genes, the eye color gene and the wing size gene, they are located very far away from each other on the same chromosome. So this is what this picture explains, right? When the genes are closer to each other, the recombinant types will be less in percentage. When the genes are further away from each other, the recombinant types will be more in percentage. The recombinant frequency will be more. I hope you understood how to explain this particular picture. This is very important. You have to observe, you have to carefully study this particular picture and try to understand what this picture is trying to convey, what concept this particular picture is trying to convey to you. Now, based upon this particular concept, Alfred Studewant was a student of Thomas Hunt Morgan. So based upon the concept of recombination and gene linkage, he came up with the concept of gene mapping, with the idea of gene mapping. He said that the frequency of recombination is equal to the measure of distance between gene pairs. The frequency of recombination is the measure of distance between gene pairs. Right? So what does it mean? It means that if the recombination frequency is more, if the percentage of recombinant types is more, that means what happens? The distance between the gene pair will be more on the same chromosome. If the distance between the gene pair is less, the frequency of the recombination will be less. So he used this idea for the purpose of mapping the genes on the chromosome. We all know that there are less chromosome number compared to the gene number. There are many genes, right? We have lots and lots of genes, but we have only 23 pairs of chromosomes. So how those genes are located means each chromosome must be carrying many genes on it. So the chromosomes, they have got lots of linkage groups. So how will you map them? We can map them by looking at their frequency of recombination. If the frequency of recombination is less, that means the distance between two genes is less. If the frequency of recombination is more, that means the distance between the two genes will be more. So that is the reason. That is the application basis of the linkage and recombination that is used by Alfred Studewant for gene mapping. So that is all for this particular video. In the next video, we will discuss about polygenic inheritance and pleiotrophy. Thank you.